Well, I love how hands-on and direct it is. Every type of science is important, from bench science to, and this is more translational, patient-directed research. And I'm really interested and excited by making something that real people could actually use in the next months or so. So that is a part of why I work for Dr. Bucher, why I work with this lab, and why I work with flu vaccine research. I like how just practical and hands-on it is. It's a pretty cool job.
Oh, yeah, Linda. Yay. Oh, I think it's, it's the virus. The virus is changing all the time. The new developments, the research, the science, the interaction with other scientists. The students are fantastic today.
All right. There's a walk-in incubator. It's really morbid here, and this is how we know where to inject the egg. This is an actual embryonated egg. So this is actually the little chicken embryo. Then you get all the little blood vessels. Okay. This is the yolk here. Okay. And this is the albumin is down here. And then this kind of pale pink peach part, that's where the egg white is. So the cells that line that cavity is where the virus actually replicates. And when the virus is released from the cells, it's released into the egg white.
We are developing strains for the flu vaccine. Most of the doses of the flu vaccine are made from virus grown in eggs. What we do is take a human virus isolates that don't grow very well in eggs, and we make them grow well in eggs. We get them to share genes with this very old virus that has been growing in eggs since 1934. We go through a series of steps when they're making the reassortants. So the first cycle, we're injecting the old virus and the current, the important virus into the same egg. And then we allow them to multiply, replicate, share genes, and so that's the first cycle. Each one of those cycles takes about 2 days. Then that allantoic fluid is pulled out. So we get rid of any virus that has the wrong surface antigens, that has the 1934 surface antigens. So we are using antibodies which have been developed against the surface antigens of the 1934 virus to get rid of any virus that doesn't have the current antigens on the surface. So we take that through a few cycles. The viruses that grow the best will outgrow the others. So after another 2-3 cycles, so each time it's 2 days, we'll end up with a population of viruses that will have the correct surface antigens and have good growth properties. And then from that, we then will clone the virus. We'll dilute it out as far as we can and find out so we can have a pure population of the reassortant virus. So these are all, so these are human experiments, H1, H2, H3. We've had, the deadly one is H5N1. And then there's also H7. This excitement was 2009. We weren't sure how bad it was going to be, and it was pretty bad. So we felt we're playing such an important role here. We know how to make these reassortants. We had the reagents available to make the reassortants. We knew that we could analyze it to get a good one out. And, unfortunately, it all worked out very well for us and for everybody else. This is what makes flu so interesting. We never know what's going to happen next.